Wikipedia. You can go to Wikipedia and Google streptococcus groups and you'll get up a lot of different stuff. Or you can read the chapter in your textbook that's called Pathogenic Gram-Positive Bacteria. And forget the number, it's on your schedule. It's in there. So, actually, there's a picture. Of hemolysis in one of those PowerPoints. Let's see if it's in this one. Of course not. Everything but a picture. I don't remember what chapter it is, so let's Google it. I think it's chapter 6. The chapter 6 PowerPoints it might be chapter 6. Blood auger. Let's see. Why can't I get it in there? What's going on? Why don't it go in the search bar? Computer is so screwed up.
That's so weird.
I know, isn't that weird? What the hell? Did I search for Ida B. Wells? She used to be here.
It's not typing.
I'm not doing anything. It won't take what I'm typing.
Oh.
This is why I end up drawing on the board. This computers are so freaking weird. Now where the hell was I? Alright, there's colors in this figure for a reason. If you have a plate of blood agar, in chapter 6, they consider blood agar a type of special media. In fact, they consider it a type of differential media. Blood agar is a Petri plate and it's red. It looks like blood. Do you know what chocolate agar is? Looks like chocolate pudding, but it ain't chocolate pudding, it's dried blood. So they also have chocolate agar. They even call it chocolate agar. What it is dried blood agar. And it's not human blood, it's usually sheep's blood. Alright, so blood agar, if you were to ask me, I would call it an enriched medium. Because you've enriched the agar with blood so that pathogens that need blood can grow. And in fact, when you're trying to find a pathogen, they'll often culture on blood agar to get it to grow. Remember, that's one of the hardest things in Koch's postulates. I was reviewing your quizzes. I had the same question on 3 of your quizzes. And it's that stupid question about an etiologist is trying to study the cause of a disease. What's the first thing he has to do? He has to try to culture the thing. So if your patient has a disease and he thinks he sees like an infection in his muscle tissue, let's say, he can't study it if he can't get it out of the patient and onto a Petri plate. And that's the hardest part of Koch's postulates. If we don't know what it is, how do we know what it needs to grow? How are we going to grow it? And in many cases, we can't. So it's like, how am I going to do Koch's postulates if I can't even get it out of my patient and onto a Petri plate to study it? So the first thing you have to do is culture the infection from your patient. Then you can start Koch's postulates. Anyway, so let's say here's a plate of blood agar. I call it an enriched medium because it's enriched with blood. Your textbook calls it differential medium because it can be used to differentiate different types of streptococcus. Let's say we have a colony of streptococcus here, and a colony of streptococcus here, and a colony of streptococcus here. We don't know which one is which. But if the agar is unchanged, it did not use the blood, then it's no hemolysis. Gamma hemolysis is no hemolysis. I'm like, then why are we even talking about it? I don't know. This would be your group D streptococcus, which we don't call group D streptococcus anymore. Now we call them enterococcus, like Enterococcus faecalis, Enterococcus faecium. Those were on our list, our escape pathogens. Why? Because some of them have developed resistance to vancomycin. But look at them. They're not even hemolytic. They're not that pathogenic. They're not that harmful. If you're hemolytic, that's worse because you're going to be breaking down your patient's blood cells. If you get a green area around your colony, you know how when you have a bruise, at first it's like really bright purple, and then as it starts to go away, it turns greenish and then yellowish, right? Mm-hmm. So this greenish color, that's why it's green, is partial hemolysis, which is alpha hemolysis. This is the symbol for alpha. Gamma hemolysis is no hemolysis. Gamma is a Y. So it's no hemolysis. This is going to be things that maybe, over here, that maybe mutans, sanguis, remember those were oral flora? They might cause a tooth abscess, but it's probably not going to be much worse than that. Streptococcus pneumoniae is also in this group. Gets in your lungs, causes pneumonia. I have those two down here. Here's your gamma hemolytic enterococcus, group D strep, now called enterococcus. They can cause infections, but they're not particularly virulent. Enterococcus is usually naturally resistant to penicillin, so that's why we had to use vancomycin, and then they became resistant to vancomycin. Alpha hemolytic streptococcus, group C streptococcus, or Streptococcus pneumoniae, causes pneumonia, which can be bad, right? But it's only alpha hemolytic. And Streptococcus viridans group. Viridans is not a genus name, it's the name of a group. That's why it's capitalized. And in that group are your oral flora, Streptococcus mutans, Streptococcus sanguis. They're normally commensal bacteria, but they can cause, you ever have a tooth abscess, you get a toothache, and your whole face swells up, and there's pus in there. Because remember, staph and strep are pussy. Now we get to the bad guys. Here's your beta hemolytic streptococcus. If these bacteria are beta hemolytic, they're going to completely digest the blood in the agar around them, and you're going to get a clear area around them. So a clear area is the worst. Beta hemolytic is the worst. Complete digestion of red blood cells. Most pathogenic. The hardest thing for people to remember, because it's confusing as hell, is that both group A streptococcus and group B streptococcus are beta hemolytic. Because you're thinking B for beta, wouldn't that be group B strep? Yeah, but it's also group A strep. They're both beta hemolytic. Group A strep is the species Streptococcus pyogenes. And Streptococcus pyogenes is gas. We call it gas, or GAS. It is commensal flora. Can also cause strep throat, rheumatic fever, scarlet fever, even necrotizing fasciitis. If you Google this stuff, it'll tell you a whole bunch of different diseases. Your group B strep, that's the one I said my girlfriend lost her baby to. She miscarried. Agalactae, strep B infections in pregnancy and in newborn infants. Okay, streptococcus is naturally usually sensitive to penicillin because it's got that thick gram-positive cell wall. But remember, they've developed resistant strains. So this is an overview of it. And then I tried to specify in here a little bit. Where do you think these names come from? Group A streptococcus and group B streptococcus. There's a poster upstairs in lab, and it tells you it comes from Rebecca Lansfield. We'll watch watch a little video about Rebecca Lansfield. I might have to, oops, no, that's not it. Where's my video? Oh, here it is. Kinda out of season.
Question. Can I count on booze to kill the bugs in my homemade eggnog? Last year we found the perfect lab to look into it. This year, holiday flair is a little sparser on the 8th floor of the Bronx building at Rockefeller University.
The university is cutting back on decoration.
But the interest in eggnog persists. Here's microbiologist Vince Fischetti.
It's probably over 40 years now. We make it every year. The recipe comes originally from Rebecca Lansfield, who is a famous scientist in this laboratory. She initiated it, making it in the laboratory before Thanksgiving, and let it sit in the refrigerator until Christmas, and we all enjoy it for Christmas. It gets very smooth at Christmas time.
So that means raw eggs have sat in this pot, in this fridge, for 6 weeks. And then everyone drinks it. And to date, no one has gotten sick. Can science explain why?
Well, the question we're really trying to answer is, if an egg were contaminated with salmonella, does the process of preparing the eggnog kill those salmonella in the egg?
To find out, Fischetti and another researcher, Raymond Shook, are running an experiment, starting with making a test batch of nog, same as they make every year, except with an extra ingredient, salmonella.
Dozen eggs.
The salmonella gets added into the eggs to mimic what would happen if you got a real bad egg.
And we're going to add sort of the equivalent number of bacteria from